All right. So I think we'll get started. It's 3:30. So I'm absconding the introduction duties today to take the opportunity to introduce my longstanding colleague and great friend, Al Spada, who's here to talk to us today about Huntington's disease. So for those of you who don't know Al, he obtained MD Ph.D. from the University of Pennsylvania, during which time he identified the first polyglutamine repeat in the androgen receptor, identifying the genetic mechanism for spinal bulbar muscular atrophy. And then he came here to the University of Washington to train in laboratory medicine, where he then developed a career in the study of multiple polyglutamine disorders, including Huntington's disease, which he's going to talk to us about today. And during that time, Al and I worked together on lots of different things, not so much Huntington's disease, and have continued to collaborate since he was recruited away to the University of California, San Diego, where he is the chief of the Division of Genetics in the Department of Pediatrics, and has continued to produce many high-profile publications in the polyglutamine field, also works somewhat in ALS, Parkinson's, and was recently, while he was here, he was a Beeson Scholar and was recently named a Harrington Scholar, and I don't know, many other awards. So, and he's going to talk to us today at my request about Huntington's disease. So thank you, Al, very much.
Yeah, great. Well, thank you, Gwen, for that warm introduction, and it's wonderful to be back here in Seattle, where I spent 16 years of my life, and they were all very pleasant ones, and nice to see some familiar faces. It's certainly a pleasure to be back here in K069, giving neurology grand rounds. So as Gwen indicated, the topic of my talk today is going to be Huntington's disease, and I'm going to tell you about work that started actually here in Seattle over 12 years ago and continues to this very moment, and give you the very latest results that will soon be published in a few months, but are not out yet. So I think that should be great fun. And I'm also, if you're not sick of me after today's lecture, you have the option of coming to hear me tomorrow for the medical genetics lecture series, where I'll primarily talk about SBMA and SCA7, and just probably throw in a little bit of the Huntington's disease stuff at the end, because I think it's really very exciting. Hopefully you'll agree with that assessment. Just to give a broad overview, I think Gwen has sort of indicated that I became interested in the polyglutamine repeat diseases some time ago, and our research program is focused on 3 polyglutamine repeat disorders, spinal and bulbar muscular atrophy, SBMA, which is a motor neuron disease, and that led us to become interested in ALS4. It's an interesting juvenile onset form of ALS that we're working on. As a postdoc here, I became interested in spinocerebellar ataxia type 7, SCA7, a cerebellar and retinal degeneration disorder, and that led us to start studying this Purkinje cell degeneration mouse model. And then, as I will explain to you today, we started working on Huntington's disease when I was an assistant professor here, a striatal degeneration disorder, and that's led us to become interested a bit in Parkinson's disease. And today's presentation will emphasize the translational aspect of our work, but if you want to really sum up what we're doing in my lab, we're using the tools of functional genomics to model disease processes, to dissect mechanisms, and to determine pathways, in the hope that those pathways will yield targets that will be amenable for therapy development. Okay, so a long time ago, when I was an MD PhD student with Kurt Fischbeck, I became interested in X-linked spinal and bulbar muscular atrophy, also known as SBMA or Kennedy's disease. And this is a lower motor neuron disease that affects only males. It's characterized by proximal muscle weakness, bulbar weakness, atrophy, and fasciculations. There are no upper motor neuron findings, which allows us to distinguish it from ALS, though occasionally patients with this disorder do get misdiagnosed with ALS. It's a slowly progressive degenerative disorder, and it has a very interesting clinical feature in that these patients have signs of androgen insensitivity including gynecomastia, which you can, I think, appreciate in this patient. I can control the pointer here. And testicular atrophy, as well as reduced fertility. And so when I was working with Kurt Fischbeck as an MD Ph.D. student, my project was to identify the genetic defect responsible for this disorder, and I was fortunate to be able to do so. What we discovered is that patients with this disease have a CAG repeat expansion in the first coding exon of the androgen receptor gene. And what we now know, and this will apply to many of the repeat expansion diseases in the field, is that the repeats tend to be polymorphic in normals. But once you cross a certain length threshold, it's about 36 in this disorder, and then you have a disease-causing repeat. And this is an absolute association for the vast majority of these disorders. The only way, to our knowledge, that patients can get SBMA is by having this particular specific mutation in their androgen receptor gene. So you have this sort of bimodal, non-overlapping distribution. And so that work was done nearly 25 years ago. And in the intervening 25 years, it's become quite clear that repeat expansion mutations are an important cause of inherited neurological disorders. And you have a very lengthy list here, and you'll see some disorders on here that are quite rare that you perhaps have never heard of, like dentatorubral pallidoluysian atrophy, and then there'll be other disorders that are much more common that you probably have heard of and probably have even cared for patients with some of these disorders. And don't be misled by this frontotemporal dementia amyotrophic lateral sclerosis chromosome 9, because this repeat, the C9orf72 G4C2 repeat, has turned out to be the most common cause of familial ALS, as perhaps many of you know. So the field is still quite vibrant, even though it's more than a quarter of a century old now, just about. But as was alluded to at the outset of the lecture, I've primarily been interested in the CAG polyglutamine repeat diseases, of which there are 9: SBMA, Huntington's disease, dentatorubral pallidoluysian atrophy, and spinocerebellar ataxias. 1, 2, 3, 6, 7, and 17 comprise this category. And these disorders share a number of features. They all obviously affect the nervous system. They're dominant mutations with the exception of SBMA, which is X-linked. They show comparable median ages of onset. They're all slowly progressive disorders, and they all show this correlation between increasing expansion size and disease severity. And it's this correlation plus the tendency of the repeats to expand as they're transmitted from parent to child that explains this very interesting phenomenon of anticipation, which was actually described more than 100 years ago by a German physician who was studying patients with myotonic dystrophy. And all of these disorders are caused exclusively by CAG repeat expansion mutations and their respective disease genes, with the exception of SCA6, that admittedly perhaps is an oddball member of the category. Okay, so in these diseases, patients have too many CAG repeats. They end up making a protein that has too many glutamines. Why is that bad? Well, the protein misfolds. And years of work have demonstrated that the misfolding of the protein is the crux of the pathology. It's really the initiating step in the pathogenic cascade. And for the majority of these polyglutamine disorders, the length threshold is relatively shared, with some variation, mid-30s, low 40s. Once you cross that length threshold, then the glutamine tract adopts this abnormal conformation, and you get a process of protein misfolding and the initiation of the pathogenic cascade. Now, we were working on these relatively rare polyglutamine diseases in the early mid-90s, and at that time, that work coalesced with studies that were being performed on Parkinson's disease, where it was found that an autosomal dominant pedigree segregating Parkinson's disease was attributed to mutations in the gene alpha-synuclein, and then it was found that alpha-synuclein is a core component of Lewy bodies. So about 20 years ago, the field came to a very important realization, and that is that all neurodegenerative diseases share a common link in that they all involve proteins that misfold. And they're different proteins for the different disorders, as shown on this slide. And then what happens is the different proteins misfold and they form what are called aggregates or inclusions. Some of them, have been named by great neurologists and neuropathologists of years long gone by. And this is a pattern now that we've come to appreciate as really a defining feature of all neurodegenerative disease. And this is a very important realization, because what it says to us is that protein quality control is a common problem in neurodegeneration, and if we can come up with a way to rectify it, we would have a therapeutic intervention that theoretically could be applied across a wide range of disorders. So that, I think, is really worth emphasizing. Now, there's another aspect of neurodegeneration that I also should mention that is a conundrum that those of us in this field still struggle with, and that is this question of selective vulnerability. And so on this slide, what I've and then I've shown you the parts of the nervous system that are in the different disorders. And as it turns out, for these different disease genes that cause these different disorders, you make misfolded proteins, but the misfolded proteins are expressed throughout the entire neuroaxis. In fact, if you take the example of ALS1, which is caused by mutations in superoxide dismutase 1, we know that every cell in the body expresses that mutant protein, yet those patients only suffer degeneration of motor neurons. So we don't understand the basis for this selective vulnerability in these disorders. And again, understanding this phenomenon will be very important for developing rational therapies to treat these maladies. Okay, so yes, I've tried to adapt to life down San Diego. This is off the coast of Mexico. In our business, you have to get used to swimming with the sharks. So this is my attempt to do that. But the whale sharks really are nothing to worry about, as I learned. And I would encourage people, if they go down to Mexico, to do this sort of thing. It's really quite fascinating. Okay, so we're going to talk about Huntington's disease, and I think this audience needs very little introduction to the disorder. But just very briefly, it's an autosomal dominant disorder. It's very prevalent in individuals, especially of Caucasian European ancestry. 1 in 5,000 to 1 in 10,000 is what most geneticists believe. It's a movement disorder, as you know. Patients suffer cognitive decline. They sometimes have psychiatric problems, which indeed can be the presentation of the disorder for certain patients. The onset is usually in the 4th or 5th decade of life. The disease is relentlessly progressive, and patients succumb to it 10, 20, sometimes 25 or 30 years after the onset. And it is in the midbrain, particularly in the basal ganglia, where you see a prominent degeneration with medium spiny neurons of the striatum exquisitely vulnerable. Now, another aspect of the disorder that has been underappreciated, but now we really are recognizing, is that patients have metabolic problems. These patients suffer muscle wasting and have problems gaining weight. And this isn't because they're moving around all the time. This is a true phenotype that has its own genesis. And of course, this is a CAG polyglutamine repeat disorder, and there's no treatment, let alone cure, for the disease. So when you think about the mutant huntingtin protein, you realize, as I alluded to earlier, that it also is ubiquitously expressed. All cells, or most cells, we think express it, but we have this pattern of selective cell death in the nervous system. And multiple pathways have been proposed to be involved, as this slide shows. However, I've highlighted 2 pathways because I think the data for these pathways is particularly compelling. And one that I've highlighted is mitochondrial dysfunction. And as it turns out, even before the disease gene was identified, outstanding research was being done by people in the field, including individuals like Flint Beal, who did experiments to show that if you treated rodents with mitochondrial toxins, that the medium spiny neurons of the striatum were exquisitely vulnerable. And much more work has been done on patients, imaging, and we have experts in that sort of thing here in the room who could elaborate upon it better than I can. But there's good reasons to think that mitochondrial dysfunction is an aspect of disease pathogenesis for Huntington's disease. Now, once the disease gene was identified, it turned out it encodes a protein that's very large, but that protein gets truncated down to an N-terminal fragment that has been shown to be a bona fide transcription factor. And studies have shown that access to the nucleus is required for pathology, and much literature has been generated to show that transcriptional dysregulation is also a key feature of Huntington's disease pathogenesis. So we have these two different pathways for which there's very strong data, and they appear to be disconnected. The other thing I want to emphasize is we're very fortunate in the polyglutamine field, and this would include for Huntington's disease, in that many transgenic animal models have been created that nicely reproduce key aspects of the HD phenotype. You get motor symptomatology, you get the formation of hallmark protein aggregates, muscle wasting, tremors, and premature death. So this has been very fortunate for those of us working on these disorders. Now, back when I was an assistant professor and I felt there was a good chance I was going to get promoted, I decided that I would work on Huntington's disease when I was approached by a fellow by the name of Patrick Waite, who was interested in working on Huntington's disease and wanted to do a preclinical trial. And we got some money to do that, and I thought this would be great. But I was concerned that the drug treatment that we were going to initiate was going to affect body temperature. So I asked him to record body temperature in Huntington's disease mice. And this is when we made a rather shocking discovery. And what you see here on the right, these are traces of body temperature from individual mice. And what you can appreciate is sometimes 2, even 3 weeks before the mice die, the body temperature drops to a very low level, sometimes down below 30 degrees Celsius. And you know, body temperature is very tightly regulated in mammals. So this was a bit of an unexpected finding. And on the left, you see that if we looked at the group, that the onset of hypothermia it started about the time that the mice were starting to exhibit signs of their disease. And so this was totally a serendipitous finding. And in science, I think when you make these sort of unexpected discoveries, you really need to pursue them. I should say this is the N17182Q mouse model that had been generated by David Borschelt, and in particular by Gabrielle Schilling, who I should note in passing has succumbed to Huntington's disease. She developed this mouse model, and these mice have been around for 6 years at this point, and no one had noticed this phenotype. So we decided to pursue the mechanistic basis of this. And to do that, we needed to become students of thermoregulation. And as it turns out, rodents have a way of regulating their body temperatures called adaptive thermogenesis, and it involves brown fat, which, as you can see here, there's plenty of depots of brown fat in rodents. And I should also note in passing, when we started our work on this, it was believed that humans only had brown fat when we're infants. But now it's known that we have brown fat, and there's beige fat, and there's a whole field devoted to understanding fat biology. But anyway, back then we didn't realize that. But we knew brown fat was important in rodents for regulating body temperature. That had been well established. And the way it works is shown here on the slide. Cold is sensed in the brain, and then you get a sympathetic outflow. You get the activation of beta-3 adrenergic receptors. That leads to the elaboration of an important transcription factor called PPAR gamma coactivator 1 alpha, or PGC1 alpha. Now, PGC1 alpha coactivates the expression of UCP1 uncoupling protein, and it leads to the production of greater numbers of mitochondria in brown fat cells. But you see, when UCP1 is upregulated, this leads to an uncoupling of the oxidative phosphorylation pathway. So instead of generating ATP, you engage in a futile cycle and you generate heat. And so this is how rodents thermoregulate themselves. And so we started the work in 2003. And then when we were into the work a year, a paper came out in Cell from the Spiegelman lab where mice that were knocked out for PGC1 alpha were generated. And it was found that they had metabolic abnormalities, but they also had neurological phenotypes that the authors argued were reminiscent of Huntington's disease. So we figured we must be barking up the right tree, so we forged ahead. And this work has been published a while ago, so I'll just highlight 2 of the key findings. We basically looked to see if there was interference with the transcriptional function of PGC1 alpha by using a promoter reporter. This promoter is taken from the UCP1 gene, and what we're doing is we're studying this in pre-adipocytes, so pre-fat cells, and what you see is that if you express the mutant huntingtin protein, you have a significant repression of this transcription activation, but if you co-transfect in PGC1 alpha, you can rescue this repression. So you see a Huntington polyglutamine length-dependent repression of PGC1 alpha transcription, and it can be rescued by overexpressing PGC1 alpha. So we did a number of studies to sort of characterize that in the mice and in cell culture, but I think the most compelling experiment is shown in this slide. And here, what we did is we did a microarray analysis. This is RNA that had been dissected from the brains of patients who were pre-symptomatic or very early in their Huntington's disease, and also from controls. And this comes from the caudate. And the bottom lists, you can't read this, it's not meant to be legible, but it's 26 different PGC1 alpha target genes. And what you can appreciate is that there's coordinate downregulation of PGC1 alpha target genes in the brains of these pre-symptomatic or early symptomatic Huntington's disease patients. And so that led us to conclude that PGC1 alpha transcription interference is contributing to Huntington's disease pathogenesis. And this is something that we published about 9 years ago. And when Tim Greenemeyer was writing a piece about the relevance of this for understanding Huntington's disease, he said, well, you know, I think this is important because we used to think of the mitochondrial dysfunction and the nuclear transcriptionopathy as disconnected, but perhaps the PGC1 alpha transcriptionopathy can be viewed now as a primary contributor to the mitochondrial dysfunction in Huntington's disease. Okay, so that's where we were in 2006, and then at the completion of that work, there were two questions that we really wanted to answer to move this work forward. And the first question is shown here, and that is, will genetic restoration of PGC1 alpha function ameliorate disease progression and neurodegeneration in HD transgenic mice? And this work, it was led by my former postdoc, Taiji Tsunemi, who is now an assistant professor at Northwestern University School of Medicine. And our approach was to do an inducible genetic rescue, where we obtained a transgenic mouse line from Dan Kelly that has a TET responsive element there, which will permit sort of regulated expression. And we obtained a line of mice with the reverse TET transactivator that was knocked into the ROSA26 locus. This is a promoter that will drive expression in most tissues. And the idea is if you give the mice doxycycline, which crosses the blood-brain barrier, you'll be able to activate the reverse TET transactivator, and you will turn on the expression of PGC1 alpha. So we generated mice that were biogenic, that contained this reverse TET transactivator and the TRE responsive PGC1 alpha transgene. And we did some pilot experiments, and we showed, sure enough, that if you gave doxycycline, you got nice induction of PGC1 alpha in the brain, some induction in muscle, not very much induction, unfortunately, in brown fat. And then we embarked upon a very heroic triple transgenic cross, where we took the ROSA26 RTTA TRE PGC1 alpha mice, and we crossed them with these N17182Q mice. Sometimes we had ROSA26 there just to sort of increase the likelihood of getting what we wanted. And if you do this experiment, you get there's one problem here, but I think we can deal with it. You get three different types of cohorts, and you get basically your HD mice, where there's no chance of overexpression of PGC1 alpha. You get triple transgenics, which when treated with doxycycline, you'll get upregulation of PGC1 alpha, and then you get controls that lack the Huntington's disease gene and should therefore be normal. So you generate these different cohorts of mice, and you initiate doxycycline treatment at 4 weeks of age for all of them, so the experiment's properly controlled. And what we observed is that when you induce PGC1 alpha in the HD mice, you had a rescue of motor phenotypes. This is a rotorod task, and what you see is that the treated mice perform very comparably to non-HD controls. And if we do ledge test analysis and look at their grip strength, we also see a rescue of motor function. And so this was very exciting because it validated our earlier work. But then comes the other instance of serendipity, and that is we saw something we were not at all expecting, and that is if you look in the brains of the HD mice, of course they have a lot of protein aggregates in the neuronal nuclei, but when we looked at the ones that were overexpressing PGC1 alpha, the aggregates were virtually eliminated. And that was something we did not anticipate. And of course, if you control this experiment and you don't give doxycycline, you don't see the aggregates removed. And the other important thing is that when you do neuropathology analysis, you see a rescue of neurodegeneration in that striatal volume and striatal neuron number is increased nearly up to normal. So this was very unexpected and very exciting. And so we wondered, what the basis for this might be. And so using a cell culture model where we express mutant huntingtin and we count the percentage of cells who have aggregates, we noted that PGC1 alpha obviously reduces the aggregation, but if you block the ubiquitin proteasome system with lactocystin, you no longer get a reduction in aggregates. But perhaps more tellingly, if you block with 3-methyladenine, which inhibits the autophagy pathway, not only are aggregate levels not improved, but they actually get worse. So that led us to consider the role of a pathway known as autophagy, macroautophagy, but we call it autophagy. And this is a pathway that I should just, at the outset, this is where I have to give a disclaimer. I think that autophagy is the single most fundamentally important pathway in the cell. So, if you haven't heard about the pathway until today, then, you can look back the rest of your life and say, oh, I remember that Spada lecture, he told me about autophagy. And if that's the only thing you remember, I feel my job will have been done. But, seriously, this pathway is a fascinating pathway. It's a pathway by which the cell is able to degrade macromolecules and organelles and beaten up proteins. And it's a pathway that really was worked out by studying yeast, and it involves the formation of a double membrane structure that ultimately encloses the material that's destined to be degraded in an autophagosome. And then you have fusion with a lysosome, the formation of an autolysosome, a drop in pH, the activation of the lysosomal enzymes, peptidases, and the degradation of the material contained within, which then is released back into the cell to be used again for biosynthetic processes. And in yeast, it's a survival mechanism in the face of starvation, but the pathway is conserved all the way up to us. And in multicellular organisms like ourselves, it's been adapted to do many different things. It's important in development. It's important in immune function. It is important in cancer. And it's important in the nervous system. And these two papers that came out back to back in Nature in 2006 really drive that point home, where if you take mice and you eliminate autophagy pathway function just in the brain by doing a conditional knockout, in one case ATG5, in the other case ATG7, I believe, what happens is the mice are born, they seem fine, but soon after they're weaned, they start to develop movement disorder, and they lose weight, they get really sick, and they die. And if you look at their brains are chock full of ubiquitin aggregated things that are misfolded proteins that are accumulating. And so it almost looks like the sort of pathology you would see in a neurodegenerative disorder. Take your pick. And so what this tells us is that the nervous system relies heavily on autophagy for maintaining homeostasis. So we thought this could be important for the phenomenon we were observing. And so that led us to consider a protein known as transcription factor EB. And a paper came out in 2009 that described this transcription factor as regulating lysosome biogenesis. So this caught my attention because the lysosome is part of the autophagy pathway. But then in a follow-up paper from this group, led by Andrea Bellabio, who's in Italy at TGEM, they showed that not only is this TFEB important for lysosome biogenesis, but it's also important for the autophagy pathway. And so the model that has emerged is that TFEB is a master regulator of lysosome biogenesis and autophagy. Under conditions where there's plenty of nutrients and no stress, it's phosphorylated and it's excluded from the nucleus, but under starvation or stress conditions, it becomes dephosphorylated. It goes into the nucleus and it activates the expression of genes that are important for autophagosome formation, for autophagosome lysosome fusion, and also for lysosomal biogenesis and function. And since PGC1 alpha is a transcription factor, we thought perhaps it was involved in the regulation of TFEB. So we looked in our mice and we found that TFEB levels are down, but rescued, in fact way increased when you overexpress PGC1 alpha. You see a similar result if you do this experiment using a cell culture system. And we found that PGC1 alpha occupied the promoter of the TFEB gene by doing chromatin immunoprecipitation. And then what we did is we generated a promoter reporter. And we found that in neuro2A cells, that if you overexpress PGC1 alpha, you can massively induce this TFEB promoter reporter. And then what we did is we took cells from mice that, knock-in mice that contain an expansion of the glutamine repeat in their Huntington gene. These are heterozygous knockouts. These are homozygous expansion mice. And these are striatal-like neurons that are being cultured and then being co-transfected with this reporter. And what you see is that you have a repression of TFEB promoter reporter activity in the homozygous mutant mice, but this is rescued when you overexpress PGC1 alpha. And what we finally did in this paper is we tested to see if PGC1 alpha was upstream or downstream of TFEB using this aggregation assay where we transfect in the Huntington with 104 CAG repeats into neuro2A cells. And again, when you overexpress PGC1 alpha, you see a reduction. If you overexpress TFEB, you see a reduction. If you overexpress TFEB and you knock down PGC1 alpha, you still see a reduction. But if you overexpress PGC1 alpha and you knock down TFEB, you no longer see an aggregate reduction. So that tells us that PGC1 alpha relies on TFEB to drive this anti-aggregation proteostasis and therefore is upstream of TFEB in the pathway. Okay, so then that's what we published a few years ago, and I think that this has relevance for our understanding of mitochondrial biology, because PGC1 alpha is the main factor that drives mitochondrial biogenesis. It promotes the expression of genes that encode proteins that are in the TCA cycle and in the oxidative phosphorylation pathway. So when PGC1 alpha is engaged, you're really generating lots of ATP. There's a price to pay for that, though, because sometimes you cannot run the system perfectly, in sync, and so you have an increased stress and sometimes increased production of reactive oxygen species. But as it turns out, as reported by the Spiegelman lab, PGC1 alpha also co-activates the expression of genes that encode proteins that prevent oxidative species from building up, the so-called antioxidant defense system. That makes sense. But then the other thing that happens when you drive your mitochondria fast and hard is they get beat up. So doesn't it make all the sense in the world that PGC1 alpha should drive the expression of TFEB, which promotes the autophagy lysosome pathway, which is required for mitochondrial autophagy or mitophagy to keep our mitochondria in tip-top shape. So we thought that was one way to sort of understand this result in terms of normal mitochondrial physiology. Okay, so the second, I promised you there were two questions that we were trying to answer. And so the second question is in this slide, and it is, what is the mechanistic basis of PGC1 alpha transcription interference in HD? We did not find any evidence for PGC1 alpha directly physically interacting with the Huntington protein. So we figured it must be some other transcription factor. And so what we did an unbiased screen for transcription factors that bind to the Huntington transcription factor using a transcription factor IP array. And one spot that lit up was the spot that contained the PPRE. So proteins that bind to the PPRE are what are called PPARs, and there's 3 of them. So then we looked at the three of them to see which might have a physical interaction. And the three of them are PPAR alpha, PPAR delta, and PPAR gamma. And this is a co-transfection assay in HEC293 cells. And I think what you can appreciate is that we have evidence for an interaction with PPAR delta, but not with alpha or gamma. And just very briefly, what are the PPARs? Well, the PPARs belong to a very large family of nuclear transcription factors, the so-called steroid thyroid hormone superreceptor family that has the stereotypical domain architecture shown on this slide. And they've been studied now for well over 20 years, 25 years in fact, and what we've learned about them is they're very important as metabolic regulators. PPAR gamma has been of particular interest in, it's expressed in white fat cells and in other tissues and has shown to be very important in diabetes and metabolic syndrome. PPAR alpha highly expressed in liver, very important for fat metabolism. PPAR delta also very important for metabolism and shown to be highly expressed in muscle where it favors an oxidative metabolic profile. And individuals such as Ron Evans, who's at the Salk Institute, not far from where my lab is located, has studied PPAR delta and shown that in muscle it can drive a super endurance phenotype. But as it turns out, PPAR delta is even more highly expressed in central nervous system than it is in muscle. And that really caught our attention and made us wonder about its role in Huntington's disease and its role in normal neural function. So what we did is we looked to see if PPAR delta interacts with Huntingtin protein, and we took cortex from mice, Huntington's disease mice. These are BAC-HD mice that were developed by William Yang. And if we pull down the Huntingtin protein and we immunoblot with an antibody against PPAR delta, you see evidence of an interaction in vivo. And if we do it the other way, by pulling down PPAR delta and we blot for Huntingtin, you see an interaction and you see that both the polyglutamine expanded as well as the normal Huntingtin protein interacts. And then we looked to see if there was a problem with transcription regulation. And so here again, we're using these striatal-like neurons that come in three flavors: normal, normal, normal Huntingtin expanded, and homozygous Huntingtin mutant. And you see a repression of PPAR delta transcription because here we're using a PPAR delta agonist for a PPRE reporter. And you can rescue that partially by overexpressing PPAR delta or overexpressing PGC1 alpha. If you overexpress them both together, you get a superinduction of this promoter reporter activity. And we've done a lot of work to look at the toxicity rescue by PPAR delta in primary neurons. So in these experiments, what we're doing is we're taking primary cortical neurons from BAC-HD mice or from non-transgenic littermate controls. And just the stress of growing the neurons will cause cell death, which is read out by detection of activated caspase 3. But if we and you can see there's greater cell death for BAC-HD neurons than controls. But if we treat with the PPAR delta agonist GW501516, you get a significant rescue. If you transfect in PPAR delta, you get a significant rescue. And if you do both together, you get the greatest rescue from toxicity. And we've looked at other readouts as well, but in the interest of time, I will not tell you about that. But what I will tell you about is work that we've done to look at the normal function of PPAR delta in central nervous system. And so what we've done is we've generated a mouse model where we've taken the PPAR delta cDNA and we've introduced a mutation that causes it to be a dominant negative. And we've put this in a system where you have a very strong promoter, but it's restricted by this stop cassette, which is surrounded by two LOX-P sites. So when you cross with mice that have Cre recombinase, you remove the stop cassette and that releases expression wherever you want it to be. And we are interested in nervous system, so we've used Nestin Cre mice where you'll have expression throughout the nervous system. And we've used RGS9 Cre because RGS9 Cre drives expression just in medium spiny neurons of the striatum. And this is very important, for Huntington's disease. And what we've seen is that if you express dominant negative PPAR delta throughout central nervous system, that the mice get significant phenotypes, weight loss, they have problems with motor function and with their their motor performance. And this is work that has been done with Elijah Maslia at UCSD to look at the neuropathology of these mice. But you really don't need a neuropathologist to see that this is not normal at 8 months of age to have your brain so reduced in mice that really are not that small. And if you look throughout the nervous system and you do histopathology, you see a dropout of cortical neurons. You see a decrease in MAP2 positivity. And also, I should emphasize, if you look in substantia nigra, you have a reduction in Th positive neurons as well. But I mentioned that we also looked at RGS9 Cre, and we found that just expressing dominant negative PPAR delta in medium spiny neurons was enough to produce motor phenotypes, and also, this was very significant in terms of motor performance, and also to produce frank neuropathology, where you had a dropout of striatal neurons. So it appears that PPAR delta is very important in central nervous system and in regions of the brain that are important for Huntington's disease. We've also looked at mitochondrial function, and we've seen that in the PPAR delta dominant negative mice that there is that the mitochondria become much smaller, and also you have impairment in complex 4 and complex 5 activity. And what work in 2015 would be complete without doing next generation sequencing, RNA, transcriptome analysis. So we have dissected out the striatum of the dominant negative mice and from their non-expressing controls. And I say this sort of tongue-in-cheek, but the experiment has really been quite interesting in that the gene expression alterations that you observe when you do bioinformatics analysis and you find out the pathways that are altered, and then you go back to that human array data that I showed you early on in the talk, and you do the same bioinformatics analysis and you rank the pathways are ranked almost identically, so much so that the likelihood of this occurring by chance is incredibly small. And we validated the hits by doing RT-PCR to make sure that they were accurate. So this shows the importance of PPAR delta for Huntington's disease pathogenesis. And so the conclusions for this part of the talk are that mutant huntingtin protein interacts with PPAR delta, resulting in reduced PPAR delta transactivation in the CNS. This contributes to the pathogenesis of HD. PPAR delta plays a key role in promoting normal neural function, as expression of dominant negative PPAR delta in transgenic mice is sufficient to produce dramatic HD-like neurological disease and neurodegeneration. And PPAR delta is one component of an important pathway for energy production and proteostasis, and thus represents an appealing target for therapy development for HD. And so I'd like to end by just telling you the therapy results because they're very exciting. I'll try to be very quick. But what I want to emphasize is that when you think about therapy for neurodegenerative diseases, you need to remember that neurons are very specialized cells. They first face unique challenges, and the unique challenges that they face have to deal with the fact that neurons are under constant demand for high levels of energy, and also they're on the edge for protein and organelle quality control. And these two things are inextricably linked. If you don't generate energy, you can't do quality control. And if you don't do quality control, your mitochondria will not stay healthy and happy, so you won't be able to generate high levels of energy. So it's my view that it is this pathway, the decline in this pathway that is inextricably linked, that is an opportunity for therapeutic intervention for neurodegenerative disorders. So I've told you about the importance of PPAR delta. So the question that one must ask is, do we have any promising compounds for potently and selectively agonizing PPAR delta function? And as a matter of fact, we do. We have a drug called KD3010. We won't try and pronounce its real name. It's a very selective and potent PPAR delta agonist, and importantly, it has progressed through phase 1b clinical trials. The indication was for diabetes and metabolic syndrome, so it's been shown to be safe and well tolerated in humans for at least 30 days. So we took this drug and we tested it to see if it would prevent toxicity in primary cortical neurons from the BAG-HD mice. And you can see at low nanomolar doses that it's quite effective at protecting against toxicity. And in this case, we were looking at mitochondrial membrane potential, and it was quite effective there in restoring mitochondrial membrane potential. And we can control these experiments by using the inactive enantiomer of KD3010. which does not have any neuroprotective effect. And so what we did then is we took the drug and we did pharmacodynamic studies to see if we injected it peripherally, if it would cross the brain and engage with PPAR delta targets. So we took black 6 mice and we treated them for 1 week with KD3010 either at 10 milligrams per kilogram per day or 50 milligrams per kilogram per day. And what you see is, and the vehicle-treated mice as well, and these are PPAR delta targets, and you can see at the 50 milligram per kilogram dose, you have a nice induction of these targets. This is in the cortex and the striatum. You see this result. And so we decided to do a preclinical trial. And I should point out there's been a lot of discussion in our field about what you need to do when you do a preclinical trial. A lot has been written about this, about how to make these mouse studies work, as this author points out. And so we rigorously adhered to those principles, which are to genotype all the mice prior to baseline assessment, to exclude individuals that are sort of not within your expected performance, and then to split the litter mates between experimental groups and to do gender-balanced experiments. And so, there's been a lot of criticism of preclinical trials in our field, so we tried to do this in a way that was rigorous. And we did such a trial using the 50 milligram per kilogram per day dose, and the results that we observed in the HN171 82Q mice were very compelling in that we could rescue the neurological function. We have this little neurological exam that we do on them, rescued them on the rotorod. We also rescued neuropathology, and we extended lifespan by a couple of weeks, about 16%. And so this result was very exciting. And the other thing that we did, I'm always skeptical of my own data. So I convinced Chris Ross at Hopkins to study our compound, and we sent him a few other compounds. They were labeled like ABC. And though I guess he's up to much higher numbers than we are, so he gave some different numbers, 6474, 6475. And what you see is 6475, which is KD3010, is as effective as his most effective intervention for preventing cell death. These are striatal neurons that have been derived from stem cells from Ph.D. patients, where you take skin biopsies and reprogram them into pluripotent stem cells, and then you reprogram them into medium spiny neurons, and you see the significant rescue from cell death, but you don't really see much of an effect from the inactive enantiomer in this study, as you can see here, I believe, yes. This is it. So this is also in the paper, which is going to come out, I believe, in online in November. So with that, I know I went over a little bit. I just want to thank you for your attention, but I must point out the people in the lab who are doing all the work. I also want to recognize Bryce Sofer, who's in the audience, who's been really a long-time person behind the work that we've been doing and continues to work with me, which has been really wonderful to have him involved, even though we're like 1,200 miles or 1,500 miles apart. Victor Pineda here at the University of Washington also was involved in this work. And we have a number of funders to thank, and I want to point out also our collaborators who've been involved in this work as well. And with that, I will stop, and if you have any questions, I'll try to answer them. Thank you. Yeah.
So do human patients with Huntington's disease have issues with thermoregulation?
Yeah, so I'm going to repeat the question. The question is, do human patients with Huntington's disease have issues with thermoregulation? So this is something we looked into a little bit, and we really didn't see any evidence of it. But what we do know is that patients with Huntington's disease have metabolic abnormalities. And so my interpretation is that, rodents aren't very little people, so their physiology is different, but the pathway is still altered in humans, and that's why we think that humans have difficulty gaining weight. And we also know that there's a problem in terms of HD muscle pathology as well that relates to this pathway. But to my knowledge, there's no thermoregulatory defect along the lines of what we see in rodents, which is driven primarily by their thermoregulatory physiology, which involves brown fat. And we don't use brown fat for thermoregulation. We use shivering, actually. So, so there's reasons to not be too disappointed by the fact that that phenotype isn't conserved in those patients. At least that's my interpretation. Any other questions? Tom,
If I could just comment on that question.
please. No, because you're the person who looked into this a
We've
bit.
actually, I think I've mentioned this to you before, don't know what to make of it. We have a few Huntington's patients, and in my recollection, they tend to be the ones with more severe disease, somewhat higher repeat expansions,
Okay.
who come to us in clinic in the winter months here in tank tops
Yeah.
and shorts,
Yeah.
and appear to be warm
Yeah.
in very cold weather. And it has impressed us that there's something not right about their appreciation of their body's appreciation of the outside temperature. We've never been able to pin down any change in their body temperature, and that's not a uniform experience of them all. It's just a few people, but it's always struck us as kind of interesting and odd. And I still wonder if it doesn't have something
Yeah.
to do with this in a few people.
Yeah, so I'll just restate that for those of you at the other sites. That was Tom Bird, who's one of my heroes, by the way, and who I trained under, so I'm biased perhaps. But he pointed out that we have occasionally seen Huntington's disease patients coming to our clinic in the winter, and they seem not to be bothered by the cold and comparatively underdressed. And it's as if, that the implication of that is as if they're uncoupled, and something is not right in terms of their metabolic function, which sort of, could go along the lines of this metabolic phenotype that may relate to the PPAR and PGC1 alpha pathway. And we haven't sort of figured that out specifically, but it is suggestive that something is not right with, the metabolic physiology in these individuals. Gwen.
This is the PPAR delta agonist.
Yeah.
Does it prevent the thermodysregulation of the mice near death? And
Right.
what's your hypothesis why you don't have a longer expansion of survival?
Okay, so the question is, does the PPAR delta agonist, does it affect the thermoregulatory defect in these mice? And why isn't the lifespan, why don't they just live, like a normal lifespan? Why don't we just cure the mice? So there are two really important questions that deserve to be answered separately. So we would not predict that a PPAR delta agonist that's selective, would improve the thermoregulatory defects because it involves PPAR gamma in brown fat. And then in terms of the lifespan extension, I think what we need to recognize with these N171 82Q mice is that this is a, very dramatic version of Huntington's disease, where we're not expressing the full-length protein. We're just taking not even the normal N-terminal fragment, but a highly truncated version of it that's very enriched with glutamines, and so it's a stress that is above and beyond. And so to ask for, a treatment to sort of, basically cure the mice, given the level of overexpression and the nature of the gene product is extremely truncated, just a few percent of the full coding region of the Huntington protein, I think that may be unrealistic. And so, at least in my own view, for a pharmacological agent to be able to accomplish that. So, as it turns out with these mice, you often have neuroprotection, but you don't have lifespan extension. So I'm pleased we have lifespan extension, and this is about the longest that you see from my review of the literature. So I think that this may be, I don't want to say this is as good as it gets, but I think this is pretty darn good. So with those constraints in mind, I think your expectations have to be different for this particular model.
So are you going to look in the homozygous knocking animals? Or
So, well, the
long-
next...
other long models?
Right, the next choice would be back HD mice. So the question is, use another model, and I'm into that. But we're discussing doing clinical trials with KD3010, and the venture capital group. I've tried to convince them we should do more preclinical work, and they're like, no, let's go right into people. And I'm like, okay, let's go right into people. Sure, I'm all for that. Question.
Are PPAR delta and PGC1 alpha abnormally regulated in other polyglutamine conditions, or is that particular to Huntington's and then so how?
Yeah. So this is the question is, are PPAR delta and PGC1 alpha dysregulated in other polyglutamine diseases? Is this particular to Huntington? So we think that the PPAR delta is particular to Huntington because there is this physical interaction between Huntington and PPAR delta. And I didn't show you all that data, but it's a direct interaction. And I showed you the in vivo co-immunoprecipitation. So we think that this is an important aspect of it. We've looked at other polyglutamine disease proteins to see if there was any interference, and we've seen that it appears to be specific. We've looked at the two other diseases that we work on. One thing that we've been very intrigued about that I'll mention, even though it wasn't the point of your question, is the relevance of this to another neurodegenerative disorder, and that's Parkinson's disease. And there's considerable data from the literature that PGC1 alpha transcription interference is involved in Parkinson's disease. Work that came from a gene set enrichment analysis meta study published in Science Translational Medicine in 2010 that in an unbiased fashion identified PGC1 alpha targets as being coordinately downregulated, 426 in that case, and then work from Ted Dawson implicating Paris dysregulation downstream of Parkin as affecting PGC1 alpha metabolism. And there was a very recent paper that I think just came out in the journal Neuroscience from Ted's group, going further with that study of PGC1 alpha dysregulation downstream of this Paris sort of pathology. So we're very interested in the idea that this could be relevant to Parkinson's disease. And that's where we're focusing our attention in addition to the HD work. Okay.
Thank you so much for a great talk.
Sure.